నా లింక్ ఏదో నాకు ఇవ్వండి. Shall I start? Good afternoon everyone. Today I am going to talk about hemoglobin. Yesterday, as I told you, there is this hemoglobin present. It is present inside the RBC. So it is a reddish color pigment. That is why RBCs have got the red color, because of this pigment. So it's a reddish color pigment which carries oxygen. So hemoglobin. So as the name itself says, it has got heme, the pigment heme, and the globin. Globin is nothing but a protein. So amino acids. So hemoglobin. So each hemoglobin molecule will have 4 hemes. What you can see on the PPT, that red color ones are the hemoglobin heme molecules. There are 4 heme molecules inside one hemoglobin molecule. There are 4 heme molecules, and each heme molecule contains one ferrous. It has got one ion. The ion present in this heme molecule is in the ferrous state, not the ferric state. It is in the ferrous state. So each oxygen and each ferrous state will carry one molecule of oxygen. So hemoglobin will carry 4 molecules of oxygen or 8 atoms of oxygen. The one what you can see in this red and blue color are the polypeptide chains. 2 are alpha chains and 2 are beta chains. So this spiral ring has got 2 alpha chains and 2 beta chains. The main function of it is carrying oxygen. Okay, the red one what you can see is the heme molecule. So it contains ferrous ion. That ion carries 1 molecule of oxygen each. So one hemoglobin molecule will carry 4 molecules of oxygen. So this is the structure of hemoglobin. Next, so here you need to know few names. What is oxyhemoglobin? When the hemoglobin combines with oxygen, it is called oxyhemoglobin, and it is in the name of HbO, oxyhemoglobin, HbO2. Here it is represented as HbO2 is oxyhemoglobin. And when hemoglobin combines with carbon dioxide, it is called carbaminohemoglobin. Carbaminohemoglobin is represented as HbCO2. HbCO2. And what is reduced hemoglobin? Reduced hemoglobin or deoxygenated hemoglobin are one and the same. When the hemoglobin molecule does not have any oxygen in it, it is called reduced hemoglobin or deoxygenated hemoglobin. It is represented by just the plain Hb, because there is no oxygen molecule to it. So you write it as Hb. And what is carboxyhemoglobin? When hemoglobin combines with carbon monoxide, it is called carboxyhemoglobin. Hemoglobin has 250 times more affinity to carbon monoxide than oxygen. So it is as HbCO. It is represented as HbCO. And what is methemoglobin? Methemoglobin is when the oxygenated hemoglobin or reduced hemoglobin is exposed to various drugs, or when the ferrous state in the hemoglobin molecule has converted to ferric state. That is called methemoglobin, and it is represented as HbOH. HbOH. Oxyhemoglobin is HbO2, carbaminohemoglobin is HbCO2, reduced hemoglobin is Hb, carboxyhemoglobin is HbCO, and methemoglobin is HbOH. What are the disadvantages of methemoglobin? Disadvantages are, once when the hemoglobin molecule combines with OH molecule, this methemoglobin, again it can never get attached to the oxygen molecule. It will never never combine with oxygen molecule. And this methemoglobin, when it is too much in our blood, it will give dark color. So the person will have cyanosis. So the person will have cyanosis, bluish coloration of the skin and the mucous membrane. Those are the 2 disadvantages. One is, once it has get attached to this meth compound, this methemoglobin again cannot reversibly combine with oxygen. First disadvantage. And the second disadvantage is this compound is dark in color. So when the red RBC carrying this compound is there, it is very dark and our body looks bluish in color. These are the disadvantages of methemoglobin. What are the normal values of hemoglobin? At birth, the hemoglobin is about 23 gram %. So whenever baby is born, RBCs are very high. That is why more the RBCs, more amount of the hemoglobin is 23 gram %. But when the baby is after 3 months, that hemoglobin reduces to 10.5 gram %. Why? Because first 3 months baby is fed on milk. Milk does not have any iron in it. So it becomes reduced to 10.5 gram %. Adults, the hemoglobin in males is between 14 to 18 gram %, and in females it is between 12 to 15.5 gram %. So 1 gram of hemoglobin, when it is fully saturated, will combine with 1.34 ml of oxygen. So what is fully saturated means when it is attached to, when it has got 4 oxygen molecules to it. That is why when it is bound to 4 oxygen molecules, we say it is fully saturated. So 1 gram of hemoglobin, when it has got 4 oxygen molecules to it, it will carry 1.34 ml of oxygen. So when do you say we have got 100% hemoglobin? When our hemoglobin is 14.78%, we say our hemoglobin is 100% hemoglobin. So when it is 14.78%, we say we have got 100% hemoglobin. So 1 gram of hemoglobin, when fully saturated with oxygen, carries 1.34 ml of oxygen. This you should remember. So each gram will carry 1.34 ml of oxygen. What are the functions of hemoglobin? Hemoglobin transport of oxygen from the lungs to the tissues, and it will transport carbon dioxide from the tissues to the lungs. The important function of hemoglobin is it will carry oxygen. From where? From the lungs to all the tissues it will give away. Again, it will collect back the carbon dioxide from the tissues and gets back to the lungs. Again, where it is oxygenated. Where this carbon dioxide is removed and oxygen is present. And the other function of this hemoglobin is it is an excellent acid-base buffer. Being a protein, it is an excellent acid-base buffer. Okay. So functions of hemoglobin are it carries oxygen from lungs to the tissue and it carries carbon dioxide from the tissues to the lungs. It is an excellent acid-base buffer because hemoglobin, I said globin is a protein. So it is a good acid-base buffer. Synthesis. For the synthesis of hemoglobin, what do we need? We require proteins because I said globin chains. Hemoglobin, globin is nothing but protein. For making of this globin chains, we need proteins. We need vitamins. What vitamins do we need? We need vitamin C, B12, folic acid. What are the minerals? The basic important mineral is iron. So we need iron. We also need copper, cobalt, calcium. All these three is copper, cobalt and calcium are very important for the absorption, utilization of this hemoglobin. So what will happen to the hemoglobin? How hemoglobin is broken down? Because as you all know, RBCs live for 120 days. After 120 days, the RBCs are taken up by the reticuloendothelial system or tissue macrophage system. So they will eat away this RBCs. So this RBC is broken down into heme and the globin. Because RBCs have got hemoglobin in it. They are broken down into heme and the globin. And this globin is again reused. It is sent to the amino acid pool. And this heme is oxidized to iron ferrous part. Ferrous. So this ferrous part is again reused for the synthesis of the hemoglobin. Because for synthesis we need iron. So again oxidized to heme part. heme part will get converted into biliverdin and bilirubin, and it is excreted in the bile, and as urobilinogen or starcobilinogen, it will come out in the feces and the urine. So it is catabolized first into heme and globin. Globin is reused for the used in the amino acid pool. Heme is broken down into iron ferrostate, and the remaining oxidized part is synthesized into biliverdin and bilirubin. What are the varieties of hemoglobin? Two varieties of hemoglobin are very important. One is adult hemoglobin. That is why adult hemoglobin, I said it has got 2 alpha chains and 2 beta chains. That is why adult hemoglobin is written as alpha 2 and beta 2. Whereas fetal hemoglobin has, instead of beta chains, alpha 2 chains and gamma 2 chains are there in the fetal hemoglobin. Fetal hemoglobin has more affinity for oxygen than the adult hemoglobin, and the lifespan of the fetal hemoglobin is 80 days. Adult hemoglobin lifespan is 120 days. Fetal hemoglobin lifespan is 80 days. And after the baby is born, for first 2 to 3 months after birth, the baby will have fetal hemoglobin, alpha 2 and gamma 2 chains. After 3 months, slowly this fetal hemoglobin is replaced by the adult hemoglobin. So, if adult hemoglobin is not replaced by the adult hemoglobin by the end of 6 months, we should get a doubt, because either alpha chains are not getting produced or beta chains are not getting produced. So, we should get a doubt. So, if alpha chains are not getting produced, it will lead to alpha thalassemia. If beta chains are not getting produced, it will lead to beta thalassemia. So, by 6 months, if the fetal hemoglobin chains are not replaced by the adult hemoglobin, one should get a doubt that the child or the newborn is suffering from thalassemia. That thalassemia could be alpha thalassemia or beta thalassemia. If alpha chains, it is not replaced by alpha chains, it is called alpha thalassemia, or it is not replaced by beta chains, it will lead to beta thalassemia. Next, your curriculum also says to show the hemoglobin estimation. So, how in the laboratory, how is this hemoglobin estimated? So, hemoglobin is estimated by this method called Sally's method. The one what we are showing there is comparator, and the one in the small bottle has got n by 10 HCl. So, it is a comparator, and that is an n by 10 HCl. So, what you should do, what is the principle? This method is called Sally's acid hematin method. So, the blood what we are taking, we are pricking our finger. We are taking the blood through a pipette and putting into one tube. And in that tube, we are also adding n by 10 HCl. After some time, 8 to 10 minutes, the blood, n by 10 HCl reacts with the blood, and the RBCs will liberate out the hemoglobin. So, the blood is mixed with the acid solution, so that hemoglobin is converted into brown colored one. It is called acid hematin. Acid hematin. So, you slowly dilute this acid hematin with distilled water. Dilute with distilled water till this brown color matches that of brown colored glass standard. So, here this brown color one we can see. So, after acid hematin, you leave it for 8 minutes. Once the acid hematin is formed, you keep on adding the distilled water and you check with the color onto the sides till it matches the exact color. So, that is your hemoglobin. So you dilute it and you read the HB value from that scale that has got some scale on it. It says 1, 2, 3, 4, 5, 6, 7, 8 like that. So, whenever you match with the side colored, that is your hemoglobin. That is Sally's acid hematin method. What are the equipment we need for this? But Sally's hemoglobinometer. One is the Sally's pipette. So, in that one, only one marking is there, 0.2 ml. So, you take the blood only up to that marking. It has got, that pipette has got only one marking. So, you take the blood till that marking. And it has got a stirrer. Why stirrer? Because once the acid and this blood reacts, it will liberate out some solution. Then you keep on adding the distilled water to it and you stir it with a stirrer, and you have got a pipette. In that pipette you are taking the blood, and from that blood you are putting into the tube. And what are the reagents you are using? N/10 HCl and then distilled water. So N/10 HCl should be taken, N/10 to the lowest bottom level, and the pipette has got only one marking till where you should take the blood, and you keep on adding. Wait for 8 minutes. That is very important, because the solution what we have taken will take 8 minutes to completely liberate the hemoglobin. So this is the method. So these are the apparatus. This is the Salis hemoglobinometer full set. It will come in a white box. One is the brush to clean your tubes. One is the dropper, where this one is the dropper where you can add N/10 HCl. The other one is the pipette. The center one is the tube here. Center one tube. You add first N/10 HCl into the tube. Then you take a pipette and add the dropping blood into that pipette. Wait for 8 minutes. Dilute it with distilled water, and you keep on adding the distilled water till you get this color. Okay. So what's the method? Place N/10 HCl into HP tube up to 2 grams. N/10 HCl you take up to 2 grams. That is the bottom marking on the tube. You take up to 2 grams. Then blood sample in the Salis you are taking up to 0.2 microliter. Then add blood sample to the acid solution. Then mix it with the stirrer. Allow it to stand for 8 to 10 minutes. And add distilled water drop by drop till the color of the solution matches with that of the standard tube. That's the standard tubes onto the sides. Take the reading of the lower meniscus from the graduated tube in grams. What are the advantages of this method? It is easy to perform. Supposing you are going for one survey, lot of pregnant women are there. This is an easy method. So you just can carry it in hand with you, this equipment. Then you can, it is easy to perform. You don't need expensive reagents in it. It is easy to perform. You can finish the whole experiment in 10 minutes, and it is very quick. It will not take very long. It is inexpensive, not very expensive, because we are only adding N/10 HCl, and the distilled water can be used as a bedside procedure also. Does not require technical expertise. No technical expertise is needed. It is a simple method. Anybody can do it. So what are the disadvantages of this method? For maximum color, longer time is required. Perfect matching with brown glass standard is not possible. Sometimes we don't get the class, either it will become too dark or too light. So perfect matching is not possible. And carboxy and methemoglobin, I told you what is carboxy hemoglobin, I told you what methemoglobin. And sulf-hemoglobin are not converted to acid emetin. If our hemoglobin has got, our blood has got carboxy hemoglobin or methemoglobin and sulf-hemoglobin, we can't convert that into acid emetin. Only when our blood has got mixed with oxygen, only we can do this procedure. And developed of color is low, and acid emetin is not stable. If you leave it for some time, the color will fade away, and also the color of the tubes also might fade away. If you keep it in sunlight for over a long period, the color of the tubes also will fade away. So source of light will influence the comparison of colors. And when we are matching with the side tubes, if we are seeing it in the dark room or a room with a curtain, color is different, and if you go to a sunlight, the color might be different. So depending upon the tube light or the natural light, the color may vary. So these are the disadvantages. I'm done with hemoglobin. Tomorrow I will do with the different blood cells, RBCs, WBCs and platelets. Thank you.